So I'd love to start off our session and my presentation as well by thanking Equalize, specifically Osage University partners, and then my home university, Washington University, for sponsoring this wonderful program. And I think from me personally for the opportunity to participate in this program. I would also like to thank my mentor, Jay Parrish, who's been really terrific over the past handful of months in providing me guidance, not only on this presentation, but I think more broadly in how to advance my company interest. So as Kristen said, I'm Julie Wardenburg. I'm a Donald Strominger Professor of Pediatrics and the Chief of Pediatric Critical Care at Washington University and the founder of Forward Defense. Forward Defense has a very singular goal, which is to generate a successful preventive against Staph aureus infection. And I think the COVID-19 pandemic has really put in the forefront of all of our minds the importance of infectious disease and the grip that infection has on human life. And this threat is something that as a pediatric intensive care physician, I see every day, and which is why I've spent many years of my career focusing on Staph aureus infection. So Staph aureus, which is shown on the right-hand side of the slide, is a very small bacteria, less than the thickness of a single human hair. This organism is really remarkable in that it can infect any tissue in the human body, most commonly skin infection, bloodstream infection, and pneumonia. And interesting to Staph aureus, most of the population has seen this bug. Every single one of us will have an immune response to this organism. And 30% even of us in this room right now actually have this bug on our skin or in our nose. So it's tempting to say, why don't we just ensure that we have antibiotics that can protect against infection caused by Staph aureus? But this bug is one of the leading drug-resistant superbugs. This organism has been a prominent superbug since the 1950s. So the problem that Staph aureus poses has been very well codified, especially within the United States, impacting both the community and the healthcare environment. So I'd just like to share with you a little bit of that impact. Looking at individuals like us who are in the community who present with new infection, the toll of Staph aureus has an economic impact of $1.1 billion in the United States alone. This bug is the single leading cause of skin infection and also a leading cause of pneumonia and bloodstream infection among a generally healthy population. 14 million care visits per year, hundreds of thousands of hospitalizations, of which about a quarter of them actually involve life-threatening infections. And it's been estimated that the total societal costs of Staph aureus infection approximate $13 billion per year. Then when we look within the healthcare domain, specifically individuals who harbor unique risk for Staph aureus infection based on having surgery, Staph aureus is the #1 cause of infection. Staph aureus infections can be associated with up to a 60% mortality, which is really frightening, especially within the context of ICU patients. The economic impact has been attributed to about $30,000 per single case within the hospital. But I want to zoom out and recognize Staph aureus infection is not just a problem in the United States. This global map shows the impact of drug-resistant Staph aureus worldwide. And what you'll notice if you just look at the coloration quite broadly, yellow being at least 10% of infections caused by drug-resistant infections. And then you look at orange and even the red, and what you'll see is that up to or greater than 50% of Staph aureus cases are associated with drug-resistant disease. And this is really prominent within some of the poorest countries in the world. So the problem is not just here. This is a broad problem. So the solution that's been proposed is to develop a vaccine, and this is an obvious solution because a vaccine will afford broad protection. It obviously provides a nice opportunity to prevent disease, and it is cost effective. And multiple companies have already taken on this goal, including Merck, Pfizer, and GSK. And if you look at these approaches, each of these approaches, which I've depicted at the top of this slide, have targeted different proteins or molecules on the surface of Staph aureus, or in the case of IBT's recent vaccine, toxins that are secreted. So a broad array of toxins. The populations that have been targeted have largely been US types of populations: cardiac surgery patients, spinal surgery patients. And what's key about each of these vaccine efforts, so 3 of them have been in phase 3 clinical trials, and they have all failed, including one that was terminated early for additional risk that was seen to the vaccinated patients. So this really leaves us with a series of failures and understanding their impact on the field. And I want to ask the question, why do these failures matter? To me, these failures matter because of Simon. Simon was a little boy that when I was a young trainee in the ICU, I watched Simon die in a period of about 12 hours from overwhelming Staph aureus infection. And as I think about the problem of Staph aureus, I recognize the current vaccine programs will never protect infants and children, because every single one of these vaccines that's currently being designed is focused on protecting high-risk adult populations. So Forward Defense has generated novel insight on vaccine design through the work of my group for many years on Staph aureus alpha toxin. This toxin is a really amazing toxin. As you can see, it looks a lot like a mushroom. And if you focus on this second picture on the right, you see the toxin forms a central membrane channel, and it pokes holes in our cells. And what's really amazing about this toxin is that it is involved in all major forms of Staph aureus infection, and it is produced by all Staph aureus strains. So as a single molecule, this is a very unique target for vaccine design. Staph aureus, what we have been able to demonstrate through our studies is that while one would ordinarily like a comprehensive immune response, including a T cell and a B cell response, in the presence of the alpha toxin, this response is dampened. It shapes the nature of this response such that there are fewer T cells, there are fewer B cells. This cognate protective immune response is blunted by the toxin. So one could say, why not just vaccinate and restore the response to the type of response that's needed? But what we know about vaccines is that vaccine success is predicated on the nature of the response that's already generated. So we can't simply convert to a better approach. So the innovation that Forward Defense brings to this field is that our approach is completely different. We recognize that infants in the first month of life are exposed to Staph aureus, and therefore they are exposed to the damaging effects of the alpha toxin. So our approach is to focus on infants early in life and protect them with a toxin-focused vaccine in order to allow the natural development of their immune response and the ability to build this T-B cell partnership to generate lifelong protection. And an important corollary of this observation is that we can predict that vaccines that are targeting adult populations will fail, because they're being given in the context of an already shaped immune response that is not a comprehensive response. So we have several market-defining attributes to our vaccine approach. We have generated and studied a novel Staph aureus alpha toxin variant that has a unique mechanism of action for which we have derived patent protection in order to move forward with this approach. We're targeting a pediatric population, and this is actually a global market. I'm not looking for a vaccine that will simply protect the children that come to my ICU. I am interested in protecting against this disease worldwide. It's about 140 million children or infants per year, with typical estimates from most vaccine programs of $2 to $40 US dollars per dose per child. And our approach generates a unique opportunity for partnership with many governmental and non-governmental organizations that have been absolutely instrumental to worldwide infant vaccination, which I think is an exciting feature for me in thinking about moving this forward. Our path ahead, in the past year or so, we have really moved from completing our academic research, which has been funded by the NIAID, towards our current STTR focused approach, where we are narrowing down our candidate selection and anticipate defining a single vaccine candidate to move forward with. This will then position us for pre-IND work, for formulation toxicology and scale up, with a goal of achieving within the next 2 years the initiation of clinical trials, and by the end of the decade to be in the phase of regulatory review, approval, and hopefully initial delivery of vaccine. So our current goal is to achieve a $1.5 million seed funding approach that will enable our IND studies to get us to this pivotal goal around mid-2024 of being able to move forward with an IND. So our group is a small group. I am joined in forward defense by Kelly Tomaszewski, who has been a wonderful collaborator of mine for nearly a decade. She's the principal scientist at our company and has invested for a long time in studying Staph aureus disease and understanding approaches to vaccination. And our principal scientific advisor right now is Steve Projan, who is himself a staphylococcal biologist and has invested in many different domains in vaccine development and is a real leader and innovator in that domain. I would like to take a minute to just thank the Office of Technology Management at Washington University, who've been absolutely wonderful in helping me navigate moving this forward, and also the biogenerator, who's provided a nice home for us as we have gotten this program off the ground. So thank you again to the Equalize program for this opportunity, and I would be happy to take any questions.
Thank you so much, Juliane. We're going to have Chris Garabedian provide the first question.
Sure. Yeah, nice presentation, Juliane, and really appreciate this opportunity. So first, I would make sure, this is partly advice and then I have a question. The work that you said you've done to prove that the alpha toxin is really the key to differentiate it from the others is to elucidate that more with whatever data you have. So I think for an effective pitch, a lot of investors will be looking to really dig into that data and try to parse that out in terms of how predictive that is to be more successful and overcome, some of the failed studies that we've seen, understanding that the adult population might be the primary reason. The collaboration with non-government organizations, I think, is really critical for vaccine development, especially if you want to get it incorporated into the CDC schedule. And NIAID often does broad-based, grants. They cast a wide net, but it's often the other organizations that you mentioned that will provide the feedback, the direction, the guidance, right? And so just curious, how much interaction have you had? Have you gotten feedback from them in terms of the optimal development? And related to that, what funding do you think you need, and what is that going to achieve in terms of gating to get to the optimized lead, IND enabling? Is the funding you're looking for to get you into the clinic? So just the interactions with NGOs and kind of exactly where the funding gets you and how much you need to do that.
Yeah. Thank you, Chris, for your constructive points. I really appreciate those. To address your questions, let me take those in a couple of different in sequence. So we're right now at the point with the STTR that we have of being able to narrow down from what is 4 candidates to a single candidate. So it's my plan to narrow down to that single candidate prior to initiating those discussions with NGOs, et cetera, to move into that next stage of the developmental process. Because I think right now there's still, as you mentioned, there's more science that needs to be laid on the table in order to really concisely demonstrate that we have arrived at the best formulation and to be able to illustrate the exact mechanism of action for that novel vaccine antigen. So I would say I anticipate probably being 4 to 6 months away from doing that. And then to address your second question, what's the funding bridge that's needed? I envision, based on how we've decided to formulate this vaccine, it's a single antigen. And we intend to approach this with an alloadjuvant because I think that's actually all that's needed. This is an old school combination, so to speak. So I anticipate that we're not going to have many troubles with that early stage of development, so to speak. So I really would like to envision that this seed funding that we will approach will allow us to build our team, which is, I don't think, going to need to be a huge team, and get us to the point where we're actually ready to have an IND filed and move into a phase 1 clinical study, which I do think will be done in adults prior to pivoting and moving on to children.
Great, thank you.
Thanks. Meghana?
Yes, no, it was a great presentation. Thank you so much. I know part of, as you explained, the solution is to have these vaccines have a really, kind of broad application. And just maybe for my own education, how is the alpha toxin going to offer that broad protection? I assume staph can mutate and potentially escape. How does it offer the kind of broad range protection that you're ideally aiming for?
Yes, thank you, Megan, and I'll address that with a few points about the toxin. The toxin is exquisitely conserved on a protein and amino acid level across all Staph aureus strains. It has not demonstrated marked mutation in all of time that humans have been infected with Staph aureus. So I think it's actually an ideal target. It's encoded within the genome, and it I think from that perspective, makes it very different than some of the other targets that have been derived. In terms of the role of the toxin in disease, our laboratory has been really fundamental in demonstrating that this toxin plays a role in almost every single type of Staph aureus disease. And I didn't get into it during the pitch, but we've demonstrated on a molecular basis how the toxin works by binding to a receptor on host cells. So now that we understand that mechanism, that really positioned us to move forward with this specific vaccine approach to target what is a ubiquitous toxin that interacts very broadly with a ubiquitously expressed host receptor.
Thank you. Appreciate that.
Great. Chris Berner, you want to round us out?
Sure. Just one quick question. You actually addressed one of my questions with respect to how you would think about developing in children relative to the adult population. But one of the challenges with vaccine development in general has been the cost of developing these drugs relative to the commercial opportunity ultimately at the back end, and some of the challenges associated with pricing and the like. I know this is very early stage, but have you given much thought to how this would fit into the overall market, and how you might be thinking about addressing some of those challenges that have been difficult for a number of larger companies as they've gone through vaccine development?
Yes, thanks for your question, Chris. I share that concern, and certainly looking at the history of pediatric vaccines, even ones that have been very successful, the market, if you will, is predicated on high numbers at a low price point. I certainly, as a pediatrician, have really appreciated seeing the work of the Gates Foundation and multiple other foundations that provide the resource to really move these vaccines forward. I anticipate, although this is, I think, in a way a little bit unfavorable from a quote market perspective, I anticipate that this vaccine will need that type of support. And I don't anticipate that this is going to be a sort of blockbuster moneymaker. And I think that's a little bit of that's challenging starting out, as you mentioned, but I think for me, the commitment really lies in ensuring that we can stop this disease. And I think the history of how we've changed pediatric infectious disease provides the rational basis to keep moving forward in spite of this obstacle.
I think it goes back to the first question you were asked about NGO engagement and how critical that's going to be. And so thanks.
Thank you all.